Let us wait for a few more minutes, after which we can start with the live session. Okay, so I think we can start. So hello and welcome everyone to the live session of week 6 on the course Experimental Biochemistry, taken by Professor Shagota Dasgupta and Professor Shoumo De of IIT Kharagpur. I am Srijan Sen and today I will be taking your week 6 live session. So in today's class we would be discussing in brief about the contents which were taught to you in the week 6 video lectures, and I would be discussing a few questions with you which would be helping you in solving the assignments and taking up other questions in your exams as well. Okay, so I'm sharing my slide. I think my screen is visible to you people. Okay, that's good. So we can start with the lecture. So today we would be basically discussing about gel and gel electrophoresis, and as to how we can, in our previous lectures we had learnt about the structures of the amino acids, then the different characteristics of the amino acids, and how these amino acids are the building blocks of our proteins, which is the functional unit of our life. So we have learned a lot about the amino acids, then the proteins. Then in the previous class we also discussed about the purification of proteins. So today we would be discussing about the how to detect these proteins, in what manner, and also not only about proteins but also about how can we detect DNA, RNA, and what types of gels we can run in order to detect these biological molecules. Also, in this gel electrophoresis, we would be discussing the role of the different chemicals which we use while making these gels, and what are the actual roles of these chemical ingredients. That also we would be discussing in today's class. Okay, so moving on with my first slides. So what is this gel electrophoresis? So gel electrophoresis is a technique which is used for the separation of your DNA or your RNA, then sometimes your protein molecules, using an electric current which we are applying to the gel matrix. So this gel matrix has a certain composition, and we load our DNA or RNA or our proteins in that particular gel, and we use an electric current to uh, to actually run the gel and then detect it. So electrophoresis refers to the electromotive force. So this is very important. What does electrophoresis mean? So electrophoresis is it refers to the electromotive force which is used to move the molecules through the gel matrix. Now by placing the molecules in the wells in the gel and applying an electric current, the molecules will move through the matrix at different rates. So this is very important because there can be some larger molecules and the smaller molecules, and their moving property or the way they move will also vary. So they will move at different rates from which we can actually separate or distinguish between the different molecules and the different compounds when we load it in the gel. So what are these different rates determined by? So they are determined largely by their mass, when the charge to mass ratio of all the species is uniform. Towards the anode if negatively charged, and towards the cathode is positively charged. Now now the flow rates also depends on the charge, then your pH, everything are the factors. Not only their mass to charge ratio, the charge to mass ratio, but also their individual masses, then their individual PIs, sometimes the pH of the buffer, then you have the how well the gel or how porous the gel matrix is. So depending on that, the molecules actually move within this matrix of the gel. So this is all about the gel electrophoresis. Now coming to the principle of electrophoresis. So this principle works on the basic understanding of your Ohm's law, which states that voltage equals to current into resistance. So this is very important, as to electrophoresis works on the law or the concept of Ohm, which was given to us. Now now how we see means, there is a current, then there is a resistance, there is a voltage. Now what Ohm is actually stating is voltage equals to current into resistance. But what is the resistance here, what is the current here, and what is the voltage here, we have to understand. So we here in gel electrophoresis, we are observing the gel to be the resistor and the power which we are applying. So when people run gels, or people who have run gels will understand that the power supply actually includes your voltage as well as your current source. So the most power supplies deliver a constant current or a constant source, and hence your power equals to voltage into current. This is known to us, so it will be providing us a constant power. Okay, so this is important that here, because sometimes there might be questions as to, even when we know that Ohm's law, but what is the resistor over here, what is actually providing us the resistance, because the voltage and the current will already come from the constant power source, and we have to remember this, that the power has to be constant, otherwise there would be a difference in the movement of the different biological molecules through the gel matrix. Now there are other equipments. So this is actually regarding the equipments and how we use it. I will be coming to this in my later slides, and I will be telling you what represents what. But as of now, in the modern techniques of how the equipments are made, so red is the positive terminal of the power supply, and it is connected to the red lead of the gel apparatus. So this much amount of details are not required, but this is just for the understanding and also having good knowledge about the equipments when you would be using them in the lab. And the black lead is connected to the black or the negative terminal, which goes to the upper buffer chamber, and the red one goes to the lower buffer chamber. Now this is organized in a manner such that during the case of your vertical slab gel electrophoresis, so what is the vertical and the horizontal one? So the vertical one is actually for your proteins, whereas your horizontal ones are for your DNA, then RNA, that is for your nucleic acids. Okay, so when the black is connected to the upper buffer chamber and the red is connected to the lower buffer chamber, so for the case of vertical slab electrophoresis, in what happens in this vertical slab electrophoresis, the negatively charged proteins or the nucleic acids moves towards the positive electrodes in the lower buffer chamber. So there is a movement from the top to bottom, or from the say from the upper side to the lower side, so from towards the anionic system. So this is the actual reason why these instruments are made in this manner, but things can vary from depending on your biological molecules, also your pH, temperature. This would be dependent on how the molecules would be moving, and you can even change the electrodes to oppose the movement or just to reverse the movement of your biological molecules. But this is actually the default mode of performing an experiment. Now, when a single gel is attached to a power supply in an anionic system, the negative charge flows from the negative cathode terminal to the upper buffer chamber and into the lower buffer chamber, which is connected to the positive anode terminal to complete the circuit. So this is actually a circuit which is being completed when the current power supply moves from an anionic system to the lower chamber, and then which is connected via your positive anode, which is completing your circuit, and the resistor is your gel matrix which is placed inside. Thereby it is following your Ohm's law. Now page, what is this page? It is polyacrylamide gel electrophoresis. So page comes for the case of proteins. So polyacrylamide gel electrophoresis. So what is page? It is an anionic system because it has negatively charged SDS. So SDS is your sodium dodecyl sulfate. So what this sodium dodecyl sulfate does in the case of proteins, it actually provides a uniform negative charge to the protein. First of all, it actually denatures the protein, it linearizes the protein. If the protein is folded or unfolded, we do not know maybe, but it whatever it does, it denatures the protein and gives it a uniform negative charge. Thereby page is an anionic system. You have to remember this. And why is it an anionic system? Because of the negatively charged SDS. So proteins are separated in cationic systems as well. In these gels, the proteins are positively charged because of the very high pH of the gel buffers or the presence of cationic detergents. So what type of cationic detergents we can use? These are also important because SDS is an anionic chemical, or you can tell it's an anionic detergent, but for the cationic detergents it is CTAB. Okay, that is cetyl trimethyl ammonium bromide. So in this case what happens, proteins moves towards the negative electrode and the polarity is reversed compared to your SDS page. So this is the total principle behind this electrophoresis. So summarizing, first of all, it follows your Ohm's law and it provides a constant power supply. The gel is your resistor. The red terminus is your positive terminal and the black terminus is your negative terminal. So when this entire system connects and the gel is placed in between, it actually completes the circuit. And another thing is that when you are running a gel, that is when you are performing a polyacrylamide gel electrophoresis, so it is actually an anionic system, but there times are there when it can be a cationic system as well, depending on the chemicals which we are using, depending on the polarity, and also the pH, the PI of the buffers, the samples, etc. And so SDS is a negatively is an anionic detergent, whereas your cetyl trimethyl ammonium bromide is a cationic detergent. Now coming to a different thing, which is your agarose. So first we need to know what is agarose. So it is actually extracted from the seaweed and is a linear polymer of sugar molecules. So it's a polymer of sugar molecules and these are connected in this manner. So when we make agarose gels, so we see from this diagram, so how we make these agarose gels. So this agarose is heated in water and the agarose polymers are flexible and the mixture of agarose and the water liquid. So we put the agarose powder in water, heat it at high temperature, and then the bonds between them breaks, but again it polymerizes to give it a very solid or a gel-like structure. Now this agarose gel is used for the separation of your nucleic acid, that is mostly your DNA and RNA. So from these two diagrams, we can see that higher the concentration of agarose which you use. So mostly you would be understanding when you run agarose gels on a daily basis, you would be understanding about the consistency of these gels. So lower the concentration of your agarose which you are using, lower would be higher would be the pores, and therefore it would be more of a flexible type, but and it would be definitely less dense. But when you use higher concentration of agarose, then you would be seeing that even the flow rate is becoming very slow because the pores are very tight, and passing through that pores becomes difficult and the gel becomes a bit more dense. So when agarose and water cool, the agarose polymers forming a matrix. When this happens, the mixture becomes more solid and gel-like. The concentration of agarose determines how dense your matrix is. So lower concentration of agarose gives more pores and it's less dense of a kind of a gel, whereas your higher concentration of agarose gives small pores and the density of the gel or the matrix increases. So this is the apparatus which we use for running your agarose gel. So here, this is the place where we actually load the samples. So in this manner, we load the samples and then we run the gel. So usually it is run at around, you say, 80 to 90 volts, not more than that. So we load the samples over here, and we know that this is the positive terminal and this is the negative terminal. So what happens is that we know that DNA is negatively charged because of the phosphate backbone of the DNA, which is at neutral pH, it is negative. So when it is running through the gel, it will run towards the anode, or the it will run towards the anode, that is towards the positive terminal. Okay. Now, how we compare the DNA fragments. So the basis of comparing the DNA fragments is separating the fragments by their size. So in when we separate the fragments by their size, we can actually compare the DNA fragment. So how these DNA fragments are formed? So there are different restriction enzymes as well as, so what we do, or when we run this DNA sample. So let me tell. So for example, we want to do a PCR. So after the PCR, we want to check whether our PCR is successful or not. In that case, we run our agarose gel to check whether our product is in that particular position or not. For example, we are doing a DNA cloning, and our insert is at around the size of say 800 base pairs. Is having a size of 800 base pairs. So we run the ladder in one or the marker, the molecular weight marker in one lane, and beside that lane we load our PCR samples to check whether we have our insert or not. So after we get the PCR product, then we purify the PCR and then we perform a restriction digestion. After that, we run to check whether we also get the product in the particular location or not. So after running this or after the restriction digestion, then we can also get small fragments. But after seeing those fragments in the gel, we can also tell that our restriction digestion step has actually worked out. And then finally we perform the ligation reaction to check. Then after that we perform transformation and then we get colonies. That's a different story. But then also you can run the ligated product with your vector to check if the ligation has been completely occurred. Then you get a big band at somewhat at a higher kilobase pair. Okay, so maybe at 6,000 base pairs or 5,000 base pairs, depending on the size of your entire construct. Okay, so we compare the DNA fragments based on this size. Now, how do we separate these DNA fragments? We run it through the gel and we perform this gel electrophoresis. then we run it on a gel, and then we check where our products are coming. And so how does this gel work? So this gel works on the principle as stated before, that is your Ohm's law, where your gel is the resistor and there is a circuit connection when you connect the gel and the two electrodes. Okay, and then you go for separation. Now, the method of separating DNA in a gelatin-like material using an electric field. So, DNA is your negatively charged, and when it is in an electric field, it moves towards the positive side. So, this is known to you. So, this is your DNA, and then this is your sample, and then on running the power supply, your DNA starts moving. So, how it moves? The larger molecules actually moves slower than your smaller molecules, but everything moves towards the positive terminus because your DNA is negatively charged. So in what manner can you compare the DNA fragments? So the size of the DNA fragment actually affects how it travels, because the smaller pieces will be traveling faster and the larger pieces will be traveling slower and they will lag behind. Okay, so from here we can tell that we can determine the sizes of the different fragments, or we can tell as to which fragment is of which size. So this would help you to actually compare the different sizes of the DNA fragments. So we are separating DNA on the basis of their sizes. So this is the entire thing in brief which has been stated. We load over here and then we run the gel, and here we can see the bands. Okay, another important thing is that how we are making this gel. So we dissolve, so we have a buffer for this, which is your 1x TBE buffer, and then you actually dissolve your agarose in that particular buffer, and then heat it at high temperature. Say in a micro oven you heat it, and then you have a tray on which you plate it after putting the wells or the combs which will make your wells. Now, like when I will be telling about your SDS page about proteins, there is something called staining, because visualization, this is a visualization technique. Okay, we need to see our DNA, we need to see our RNA, we need to see our protein. So what we will be seeing, or what we will be seeing? So for this we have ethidium bromide. Okay, so ethidium bromide is an intercalating agent which intercalates inside your DNA and actually helps for the visualization. So when the gel is made, we give some amount of ethidium bromide, dissolve it in that gel-like mixture, the fluid one, and then we actually plate it in the tray, where we will be keeping it for 30 minutes or 45 minutes for solidification. Okay, after which the gel solidifies, and then we load our sample. The gel and the sample runs, and then we place it under an UV transilluminator, which is actually shown in your diagram B. It is observed under your UV transilluminator, where you can see your desired product. Therefore, we need to remember quite a few things about this agarose gel electrophoresis. The first thing is that what is this agarose and the structure of these agarose. The second thing is that it is an horizontal one, not a vertical one. So we run it in an horizontal basis. Okay, and the smaller fragments runs faster, the longer fragments runs slower, and it lags behind. And we are separating DNA based on their sizes, and we are using ETBR for visualization, because ETBR is an intercalating agent which intercalates inside your DNA and RNA, and then we observe it under UV transilluminator to see our desired product. Also, we can stain DNA using Carolina blue stains to DNA, which would also which makes it easier to see on a light viewing box, because sometimes might not be the presence of your UV transilluminator. So in that case we can use Carolina blue stains for the staining of DNA. So this is actually the summarized version of this. So this is the place where we are loading the samples and then we are running it. Then we are getting different molecules and things like that. And from this we stain it with, either we can stain it with Carolina blue or UTVR, any one of that. For viewing it under the light, we can use Carolina blue stains, or for viewing it under your UV transilluminator, then we can use ETBR. So ETBR is mostly used for observing these gels under UV transilluminator or observing your DNA. Okay. Now coming to the polyacrylamide gel electrophoresis. So this is the entire setup. This is your power supply, and this is where you have where you will be running the gel. Okay, so these two are actually the important apparatus for your protein gel separation or your SDS pitch, the polyacrylamide gel electrophoresis. From here you can see that you have the voltage, then your current and the time. So you provide a constant voltage at around say 80 or 100 volts, and then you have a constant current supply, and the time does not matter, and then you run it. Okay, and then the gel runs at a constant power supply. So therefore it is following your Ohm's law, where a constant power supply has been mentioned. So the constant power supply has to be kept in mind, and it has to be throughout when you are running your gel. And these are the different combs which we use. These are your larger plates, and these are your thin plates, which, and then you make the gel. So after which you provide the comb for making the wells in which you would be loading your samples. And this is the structure, this is the gel image of, so this lane is your marker. Then you have different protein samples which are run on the different lanes. From this you can tell that this is a good image of a gel. So what are the details of the polyacrylamide? I am coming to it, but as of now this is the apparatus, this is the power supply. Yes, this is your power supply. This is your gel running apparatus. These are your combs. These are your larger plates. These are your thin plates, and these are your another some bigger combs. And then this is your tray where you are actually making your gel or stacking your gel. And this is an vertical system. So it runs from the top to the bottom. So coming to the separation of these gels, so the term gel, it refers to the matrix which is used to contain then separate your target molecules. So in most cases your gel is a cross-linked polymer whose composition and porosity is chosen based on the specific weight and the composition of the target which we need to analyze. So the thing which we would be analyzing, depending on that your gel matrix is made, so which is a cross-linked polymer. So how tightly your gel would be, depending on that we would be making our gels, or how well we want to resolve our gel or separate resolve our proteins. So depending on that we give a basic composition and porosity to our gels. So when we are separating proteins or nucleic acids, it is composed of the different concentrations of your acrylamide and a cross-linker, so which produces some different different sized mesh networks of your polyacrylamide. And when separating a larger nucleic acid, say a few hundred bases or few kilo base pairs, then the preferred matrix would be your agarose, which I mentioned before. So in both the cases, your gel forms a solid and a porous matrix. So here your acrylamide is so this is a basic knowledge about your acrylamide, where your acrylamide is a neurotoxin, and this is actually important for forming the polyacrylamide structure, which would be actually involved in forming the pores inside your gel matrix. And I mentioned before, saccharose is composed of long unbranched chains of uncharged carbohydrates without cross-links, resulting in a gel with large pores aligned for the separation of macromolecules and macromolecular complexes. So what we do, so after your electrophoresis process is done, the molecules needs to be stained to make them visible, because this is a visualization process. So we use ethidium bromide for your nucleic acids, then we use silver and Comasi blue dyes mostly for your proteins. Okay, sorry for the interruption. Okay, so after the electrophoresis process is done, we need to visualize it. So we use, say, your ethidium bromide for your nucleic acids. Then we have the silver and the Comasi blue dyes for observing of your protein gels. Also, now what happens is that to visualize this, if the analyte molecule fluoresces under UV light, then the photograph can be taken under that. Also, we can use other systems like the autoradiogram for recording our gels or taking a picture of that in the computer. So what fluoresces is that your ethidium bromide actually fluoresces and makes the DNA visible. Otherwise, we can take it under the autoradiogram. Now, if several mixtures have been initially injected next to each other, they will run parallel in the individual lanes. Now, depending on the different molecules, each lane shows separation of the components from the original mixture as one or more distinct bands, one band per component. Now, in each lane, you will be seeing one band, and depending on what samples you have loaded, you can separate these fragments. Now, it can happen that when the separation is not proper, so this can lead to, when the separation is not proper, this can actually lead to overlapping bands, or it can appear as smears, uh, because of which you cannot actually identify your actually the what you are wanting to observe, or the fragment which you want to see, and you would be seeing it as a, what do you call, a paint. When you paint the walls, the way you paint it appears like that one. It's an unresolved type of a thing. You would be observing that. And the bands in different lanes that end up at the same distance from the top contain molecules when passed through the gels with the same speed, which usually means they are approximately of the same size. This is important. And the molecular weight size markers available contain a mixture of molecules of known sizes. If such a marker was run on one lane in the gel parallel to the unknown samples, the band observed can be compared with your molecular weight markers, and it will also help you in determining the size. Now, the distance which these fragments or these proteins or the nucleic acids travel is actually inversely proportional to the logarithm of the size of the molecule. So they have a logarithmic relationship depending on that, but it is inversely proportional. So this is also important, that the distance which the band travels is approximately inversely proportional to the logarithm of the size of the molecules. Okay. Now, proteins have different charges and different shapes, and there are chances that they do not migrate in the polyacrylamide gels at the same rates when you place it in the gel apparatus. That is, when you are placing it from the negative to the positive EMF of the sample. So they may not be running at the same speed. Now what happens is that we actually denature our proteins using your SDS or your sodium dodecyl phosphate, because they are anionic detergents, and it gives a negative charge to your protein. So the amount of SDS is relative to the size of the proteins which you use. So the resulting denatured proteins will be having a negative charge, because of which it provides us a similar charge to mass ratio, and these denatured proteins opens up and it starts acting like your long rods instead of a complex tertiary structure. And now the rate of this will be determined by the resulting SDS coated proteins migrate on the gel, and these proteins would be separating only based on their size and not its charge or the shape. So this is the for the case of proteins. Now what happens for when we are not providing SDS? So when there is no SDS in the polyacrylamide gel electrophoresis, we call that as the native gel electrophoresis. So what is this native gel electrophoresis? So it is a technique used mainly in protein electrophoresis where the proteins are not denatured. We do not denature the protein because there is no anionic or cationic detergents which will actually denature the proteins. So therefore they are separated based on their charge to mass ratio. So this is native gel electrophoresis, not an SDS page, that is SDS page. Now, this page is used for separating proteins depending on the various sizes. It can vary from 5 to 200 kilodaltons due to the uniform pore size, and these pore size is controlled by the concentration of the acrylamide and the bisacrylamide which you are using while making of your gels or while casting your gel. So a special care needs to be taken because acrylamide, as I mentioned before, is a potential neurotoxin, and it is liquid, can be available in the liquid and powdered form as well. Now the other types of gels which we do run is your agarose gel, which also can be used to separate your proteins, but they do not have a uniform pore size, but can be used for your electrophoresis of proteins which are larger than your 200 kilodaltons. So this is another drawback, or you can say an advantage, that so two things we need to keep in mind. For the page, the protein separation varies from your 5 to 200 kilodaltons, and this pore size would be depending mostly on the concentration of the acrylamide and the cross linker, which is a bisacrylamide which we are using while casting of the gel. Another type which we use is the agarose gel, where proteins, for separating proteins higher than 200 kilodaltons, but they do not have a uniform pore size. Okay, so unlike your SDS page, native gel electrophoresis do not use denaturing agents, so are separated based on the charge to mass ratio, that is differing the molecular mass and your intrinsic charge. And since the proteins remains in the native state, they may be visualized not only by the general protein staining, but can also be visualized by specific enzyme linked staining as well. So SDS page is a sodium dodecyl sulfate polyacrylamide gel electrophoresis, which is used in different aspects of biology, your molecular biology, then your forensics, genetics, to separate your proteins based on their electrophoretic mobility. Now the SDS gel electrophoresis of the samples which are having your identical charge by mass due to the binding of the SDS in fractionation by size, and it is mostly used technique in the world. So what is the procedure behind it? So the solution of the proteins to be analyzed is first mixed with your SDS. That is an anionic detergent which denatures the secondary and the tertiary and the higher level structures and the non-disulfide tertiary structures. This is very important. It only denatures the secondary and the higher level structures, but not your disulfide linked tertiary structures, and it applies a negative charge to the protein. Now without the SDS, the different proteins with similar molecular weights would migrate differently due to the differences in your mass by charge or the charge by mass ratio, as each protein has an isoelectric point and the molecular weight is actually related to the primary structure. So these thing is actually known as your native phase. That is a native polyacrylamide gel electrophoresis, where you are not using any anionic detergent, and you are running your samples and separating them based on their isoelectric point, that is your charge by mass ratio. Now, but what SDS does is that it actually solves the problem as it binds to it and unfolds the protein, because the negatively because providing a negatively charged to the amino acids in the polypeptide. Now SDS binds in a ratio of approximately 1.4 gram SDS per 1 gram of protein. This is not much important. While making of the gels, we provide 10% of SDS. So it actually gives an uniform mass charge ratio to most of the protein. And the distance of migration is directly related to the size of the protein. So this is actually the difference between how we run an SDS page and a native page. So coming to the different rules which the chemicals have in these gels, or making of these gels and while running these gels. So the first thing is that it is a polyacrylamide gel, and uh, we separate the proteins in a polyacrylamide gel based on their size and charge. So what are the different properties of this SDS gel is that it is a very thermostable gel, it is transparent, as well as it is chemically inert, and obviously it is a synthetic gel, and it can be prepared with a varying range of pore sizes. So when we vary the concentration of your acrylamide and the base acrylamide in the different ratios, or we provide the different concentration of the different linking agents, then we can actually vary the pores. So the pore size of the gel is determined by the two factors, mostly the total amount of acrylamide which is present, and the amount of the cross linkers. So here we are assuming that the total amount of acrylamide present in terms of percentage is percent T, and that is your total acrylamide bisacrylamide monomer concentration, and the amount of cross linker, that is the cross linker concentration, that is the percentage C. Now what happens is that now why is the pore size important? Because the higher the pore size, you would see that it is resolving very slow, or the movement of your protein is very slow, because of which you would be also seeing that the resolving is good. Okay, so larger your pores, you would be also seeing that your protein is moving faster and faster, but what happens is that it won't be resolving very well. So so an optimal percent T and percent C is important for your better resolving, as well as for faster separation of your proteins and visualization. So pore size actually decreases with increasing your acrylamide and bisacrylamide concentration, and with your crosslinker. So 5% C gives smallest pore size, and any increase or decrease in the percentage C increases the pore size, as the pore size with respect to C has a parabolic function with a vertex of 5% C. We do not need much of these function details or what function it actually holds, but 5% of your crosslinker concentration is sufficient to give the smallest pore size and good resolving. And if you increase it or decrease it, it has a parabolic function, so it increases the pore size. Okay, so these gels have the capacity to withstand high voltage gradients, which is actually feasible for your various staining and your destaining procedures, and can also be digested to extract your separated fractions or dried for autoradiography and for permanent recording and clicking the picture of these gels and storing them. Okay, so now, these polyacrylamide gels have these, especially these SDS ones. The SDS polyacrylamide gels have the two layers of gels. Like for the agarose gels, it's a one layer of gel. That is, it is only made of agarose. So here it is also made up of acrylamide and bisacrylamide and the cross-linking agents, but it has two layers. Your first layer is your stacking layer or your special gel. That is where you would be loading your proteins and or your samples inside the well. So that is your stacking gel portion, and below it lies your resolving gel. So while making the gel, first the resolving gel is made, kept for 30 to 45 minutes before it solidifies. Then your stacking gel is made, and then you put the comps to make the wells. So the two important layers of the polyacrylamide gel includes your stacking gel and your resolving gel. So stacking gel actually helps in the stacking of your protein and gives it a single line to your protein such that it actually stacks very well, and it's bedded on your resolving portion, after which your protein starts resolving based on their size and whatsoever components it has. And it's because when you're running an SDS page, it has a charge, so the charge to mass ratio remains the same, so you would be separating them based on their size. The next other important component is your tris, that is your tris hydroxymethyl aminomethane. So it is used as a buffer because it is an innocuous substance to most proteins. It has a pKa of 8.3 at 20 degrees centigrade, making it a very good buffer for the pH range between 7 to 9. So tris is a very important component while running your buffers, while making your buffers. One reason is it is an innocuous substance, and the next thing, it has a pK of 8.3, making it suitably good as a buffer agent while using it in the pH range of 7 to 9. Another component is your glycine. So it is used as a source of your trailing ion or a slow ion because it has a pKa of 9.69, and we see that the mobility of the glycinate, that is your negatively charged glycine, is such that the effective mobility can be set at a value below that the slowest known proteins of net negative charge in the pH range, and the minimum pH of this range is approximately 8. So it is also falling in the pH range of 7 to 9. So what this glycine is doing, it is acting as an ion which is a slow ion, so such that even the slowest known proteins can also be resolved very well. So it actually stays at the back and actually is involved in pushing or creating the gradient, which is actually involved in, or which is basically involved in the resolving of your proteins. Now coming to the other important thing, which is your acrylamide. So what is this acrylamide? It is crystalline powder. So it undergoes autopolymerization when dissolved in water, and so this process is very slow but a spontaneous one, and by which your acrylamide molecules join together by head to tail fashion, but in the presence of free radicals. Now what is providing these free radicals, I am coming to it. So in the presence of these, just only in the presence of water, this is a very slow reaction, but in the presence of your free radicals, your acrylamide molecules are also activated into free radical state. Now these activated molecules, now we know that these radicals are very uh, quick in reaction. So these activated monomers actually polymerize quickly. These radicals, this radical formed acrylamides polymerize quickly, forming a long chain polymers. Now this kind of reaction you can call as vinyl addition polymerization. So solution of this polymer chains becomes viscous but does not provide the gel because the chains simply slide over one another. Now this gel formation requires various chains together, and as mentioned before, acrylamide is a neurotoxin, and it is also essential to store this acrylamide in a cool, dark and dry place to reduce autopolymerization and hydrolysis. So what is acrylamide actually involved in? So when it dissolved in water, it polymerizes very slowly but spontaneously in a head to tail fashion, and but when you give but when it undergoes a radical formation, then the polymerization process actually enhances, and it undergoes vinyl addition polymerization, and acrylamide has to be stored in a cool and dark place, also in a dry place, to reduce autopolymerization and hydrolysis. So acrylamide is also another important candidate or an ingredient while in running your gels. So the next one is your bisacrylamide. So it is the most commonly used your cross-linking agent. So bisacrylamide is your cross-linking agent for your polyacrylamide gels, and sodium dodecyl sulfate, as mentioned, it is the denaturing agent and it provides a uniform negative charge to the protein. So when a protein is heated at 100 degrees centigrade in the presence of SDS, the detergent, actually what SDS does, it wraps around the polypeptide backbone and binds to these polypeptides in a constant weight ratio of some given quantity, gram per gram of the polypeptide. Now in this process, the intrinsic charges of the polypeptides becomes negligible because the negative charge is spread over the entire polypeptide chain contributed by your SDS. Now the polypeptides after treatment becomes opens up, denatures, and it becomes a rod-like structure, giving it a uniform charge, negative charge density, based on which we are going to separate in an SDS page. Now, as I was speaking in my previous slide about the free radicals, the activated into free radicals. So what is actually activating these acrylamide into this free radicals? So this is done by your ammonium persulfate or your APS. So APS is an initiator of gel formation. So what APS does, it actually helps in the formation of your radicals. So therefore it is acting as an enhancer or an initiator of the gel formation, speeding up your polymerization process. But these radicals are not only important, you have to also stabilize these radicals, because these radicals have very fast reactions, and what it does, it first forms, polymerizes, again breaks and forms another bond, okay, and then it again breaks and forms another bond. So we need to stabilize these bond formation or these radicals when two radicals come close to each other. So once they come close to each other, they form the bonds, and then the chapter ends over there. So we need to stabilize these radicals. So this thing is done by your TMAD, that is your N prime tetramethyl ethylene diamine. So the chemical polymerization of the acrylamide gel is used in SDS page. So it can be initiated by your ammonium persulfate and the quaternary amines, that is a TIMID. Now the rate of polymerization and the properties of the resulting gel depends on the concentration of the APS and the TIMID which you use. Now the lowest catalytic concentration that can allow polymerization in the optimal period of time should be used, and these APS and TIMID are basically used in equimolar concentration in the ring. So what TMAD does is, so the more higher concentration of TMAD you use, you would be seeing faster polymerization, but we always want slow polymerization to occur. The reason being that it will also help to actually form uniform pores throughout your gel matrix. When you go faster polymerization and provide higher so for faster, if you have to anytime make the polymerization process faster, you provide higher concentration of your timid. Okay, so then it undergoes faster polymerization, but one problem which occurs is that the pore size is actually not uniform, and then you would be seeing discrepancies when you would be visualizing your gel. Therefore, an optimal or a good knowledge about what concentration of APS and TMAT we would be using should uh, we be having at the back of our mind before we run the gel and make the gel. Okay, so uh, okay, so another time I'm repeating about this APS and TMAT. So the APS is actually the initiator of your gel formation, and it actually helps in the formation of the radicals which are involved in the polymerization process and faster polymerization. And TMAT actually helps in stabilizing these radicals for better gel mat or better polymerization process. So both of these components are very important. So what components we spoke about? The first one is your trace, which is involved in the buffer. The next one is your glycine, which is the trailing ion and helps in actually pushing the proteins forward and helps in better resolution of even the slowest known protein. Then we have spoken of the acrylamide and the bisacrylamide. Also, we have spoken about the SDS, which is our anionic detergent providing a uniform negative charge. Then we have also talked about the ammonium persulfate and timid, and how good knowledge of the concentration of these two are important in making a good gel matrix. Okay. Now, before we move to processing and visualization, we would be taking a say 3 to 5 minutes of break before we go on with the visualization process. Okay, so we can start. Okay, so we have only one slide before we move on with the solving the questions and the assignments. That's good. So these are the list of your few chemicals which you can be using while for processing and your visualization process. These are especially for your proteins. You have a lot of staining techniques and all, but these two are mostly used. So bromophenol blue is not a staining process, it is actually visualization process when you're running the gel, because you need a particular dye to actually visualize how far your gel has run. Otherwise, how you would be tracking your gel? So your bromophenol blue is actually your tracking dye, and it actually helps to tell how far your gel has run. Whereas your Comasi blue is the one which because you are tracking for the bromophenol blue, you are tracking the run of your proteins or the samples, but you are not being able to observe your proteins, what you actually need or finally need. So Comasi blue actually does is that it stains the proteins. It has your acetic acid, then it has methanol and water. So your proteins gets precipitated in the presence of this acetic acid and this methanol, and it actually forms a mark over there, a deep blue mark. So therefore this Comasi blue is being used for your staining process. Other staining processes which you can use is your silver stain and your, say, and we have ethidium bromide for when we are using it while observing your DNA and RNA. So what bromophenol blue says, so it is a universal marker dye. Proteins and nucleic acids are mostly colorless. So when subjected to electrophoresis, it is important to stop the run before they run off the gel. Now it is used as a tracking dye because it is viable in alkali and neutral pH. So the most important thing is that it is a tracking dye, and it is a small molecule, it is ionizable, and it is negatively charged above pH 4.6. So the buffer which we use has a pH of 8.2, so it hardly matters in any way, and hence moves towards the anode. So being a small molecule, it moves ahead of most of the proteins and nucleic acids, and it reaches the anodic end of the electrophoresis medium when the electrophoresis, and then the electrophoresis is stopped. Now it can bind with proteins weakly and gives a blue color, but this is not sufficient enough to get actually the bands of our proteins. For this we use Comasi Brilliant Blue. It can be R250 or G250 depending on what we are using. So it is the most popular protein stain, and it is an anionic dye which binds with proteins it is an anionic dye and it binds with proteins non-specifically. So the structure is predominantly non-polar, so it is used in methanolic solution and acetic acid. So the proteins in the gel are fixed by your acetic acid and then simultaneously stained, as I was mentioned. Then the excess dye incorporated in the gel can be removed by your destaining process with the same solution but without the dye. Okay, so it has same, and but your dye powder is not present over there. Okay, so therefore large volume of staining solution is required, approximately 10 times to the volume of the gel. So the proteins are detected here as blue bands on a clear background. Is anionic, it interferes with the staining process. Therefore your large volume of staining solution is required. Okay. And then we have other stains, silver staining, which you can also use. So these Comasi blue stainings can detect up to your 100 nanograms of proteins, but the silver stains actually can detect up to 10 to 100 times better protein. That is, it can detect up to your 1 nanogram of protein even. So even a minute amount of protein can also be detected under your silver stain. So silver stain is better, but in a daily use we use bromophenol blue and your Comasi brilliant blue. These two dyes are being basically used. So bromophenol blue is a tracking dye for tracking of the running of the electrophoresis process, the running of your proteins, such that your protein does not run off the gel, and the brilliant blue is used for the staining process. Okay. Now coming to the practice problems. So as I discussed in my in a manner, so first you would be answering, and after that I would be discussing with you the answers. Okay, so let's see. I think these are very easy questions. We won't be taking much time. Okay, let me see. So what does the first question say? So in gel electrophoresis, the gel acts as, what does the gel act as? First I would be getting your answer. We have only one people, but it's okay. Are you there? Okay. So you can try out. So you are giving me two answers, A or C. One is C. Okay, that's correct. Yes, molecular sieve is the right answer. Number two says, which is a suitable technique for separating large DNA fragments? Agarose gel electrophoresis, SDS, native, or all of the above? Okay, so I've got an answer. It is 2A. That is also right. Yes, agarose gel electrophoresis, as I was teaching, agarose gel electrophoresis is used for separating of large DNA fragments. Yes, that is correct. Okay, number three. Which of the following can be determined using SDS page? Molecular weight of a protein, pi of a protein, presence of disulfide bonds in a protein, dissociation constant of protein ligand complex. This is an MSQ, so one or more than one option may be correct. So what do we do for the case of SDS page? Three A. Yes, we definitely determine the molecular weight of a protein. Anything else? I haven't spoken about the disulfide bonds in my lecture. That is also important. So for breaking, we also uh, determine the presence of the disulfide bonds in a protein, because even when the protein gets denatured, the disulfide bonds do not actually break, because I was teaching that it breaks your secondary structures but not your tertiary, your disulfide for linking tertiary structures. So for breaking this disulfide structures, we actually need the help of beta mercaptoethanol. So this BME or beta mercaptoethanol is responsible for breaking of your disulfide bond. So only if it is an SDS page, only when it is an SDS page, then we do not have the BME. That's why it will also help to understand if there is the presence of any disulfide bonds or not. So for question number 3, A and C both are correct options. Next one is an easy question. What is the role of SDS in SDS page? It acts as a reducing agent, imparts uniform negative charge to the proteins, controls the pore size of gel matrix, or determines the pH of the gel. Yes, that is absolutely correct. It imparts an uniform negative. It opens the proteins up. It denatures the proteins. It makes the proteins a linear rod-like structure, and then it imparts a negative charge, because of which the proteins have a rod-like structure. Yes. So 4B is the right option. Okay. Moving on to the next set. Agarose is. Question number 5 says agarose is a protein, polysaccharide, DNA, lipid. So these questions are easy actually. Yes. So agarose is a polysaccharide of your carbohydrates or your sugar molecules. Okay. So 5B is right. Question number 6. Migration of protein in SDS page is determined by charge of the protein, size of the protein, shape of the protein, pi of the protein. Okay, that is also correct. B, size of the protein is correct option. Okay, now moving on to the next question. Question number 7 says the main advantage of using separate gels for stacking and resolving in SDS page is to increase the resolution of separation, increase the migration rate, to change, maintain the charge of the proteins, to retain the shape of the protein. Why do we use two different layers of gel? And why do you say so? Do you think is the option A correct? Yes, option A is correct, to increase the resolution of separation. Why do you think option A is correct? Okay, so basically it is that the ions are playing a role over here, but another thing is that has larger pore size, it separates the protein. Actually, this is not right. For the pH, which the buffer which we use for making of the stacking gel has a pH of 6.8, whereas the buffer which we use for making of the resolving gel is actually 8.8. Okay, so there is a difference of 2, a pH varying of 2, having a varying distance of 2. Okay, so the stacking gel is actually, so the proteins in the stacking gel are stacked or are sandwiched between your glycinate or your glycine and your chloride ions. So your protein is sandwiched in between these two, whereas in the resolving thing the chloride ions moves very fast. Okay, so it is actually building up the gradient, then moves your proteins and then your glycine glycinate. So because of which your proteins can resolve very well. Okay, so this resolving gel already has a pH which is a higher pH, because of which your glycine becomes glycinate. One is this reason, whereas in your stacking gel the glycine remains in the zwitterionic form, and that actually helps in the stacking of the gel or the bedding of the your samples, whereas the resolving one actually is important in building up the gradient, which actually helps in better resolution of the separation. So what is the role of APS in SDS or native page? What does APS do? To catalyze polymerization of the acrylamide. Okay, you said to cross-link acrylamide and base acrylamide to provide free radicals for cross-linking reactions. Yes, so 8C is the right option. The next question is, in SDS page, the major ions present in the buffer are tris, glycine, chloride, all of the above. No, no answer. This is an easy question. In SDS page, the major ions involved. So I have spoken about tris being involved in your buffer formation. I have spoken about glycine. Also, have spoken about chloride in my last explanation. So all of these three are involved. And where does this chloride come from? Can you tell me where does this chloride ion come from? So while we are adjusting the pH of the tris buffer, we adjust it with HCl. So from this HCl, we are actually getting the chloride ions. So from here, the chloride is also playing a role. Okay. Beta mercaptoethanol is used in SDS page 2. Break hydrogen bonds and unfold a protein, break disulfide bonds, break the peptide bonds, break multimeric proteins into monomers. I have just said, give me the right answer. What do you think would be the right answer? Beta mercaptoethanol is used in SDS page 4. Yes, it is used for breaking of disulfide bonds. So that is correct. So 10B is the right option. So 9 it was D, 10 it is B. Okay. A non-essential component of SDS page is, which is the non-essential component of SDS page? Acrylamide, TIMED, APS or beta mercaptoethanol? You do not require TIMED? It is an essential component. It is the most essential component in making of your SDS page, because if you forget to give TIMED, you should forget to actually see your gel polymerizing. So TIMED is very important. TIMED, APS, I was teaching that these are very important because TIMED actually stabilizes your radicals, and these radicals would actually be forming your bonds, which would be actually involved in the forming of the pores, because through which it would be making, you would be making your gel matrix. So TIMED is actually very important. But see, if your protein does not have any cysteines, or does not form disulfide bonds, then you need not use, then you study and find no cysteines, no disulfide bonds can be formed, so you do not require a reagent. So what is involved in breaking of your disulfide bonds? That is your beta mercaptoethanol. So the beta mercaptoethanol is not an essential component, but if you see formation of disulfide bonds in any manner, then you can provide beta mercaptoethanol. So for question number 11, it is D. Beta mercaptoethanol is the non-essential component in SDS page. Okay. The next question says, to visualize the proteins in gel, which of the following staining method is used? Comasi blue, ETBR, gram staining, silver staining. To visualize proteins in the gel. Yes, Comasi blue is right. Another one, can you see? Anything else for proteins? I was also speaking about silver staining. So your Comasi blue staining is also correct and your silver staining is also correct. So it is option A and D. Okay. The next question. These are easy questions. These are very quick questions. So you can expect some of these in your exams, as well as you have definitely solved your assignments or would be solving them. So this would always be helpful, and you would be getting very direct questions from this regarding your chemicals and the uses of these chemicals. Then how we are separating these gels, what are the components which we are using, how we are separating, what are the staining methods, this and this. Okay. Oh, before I said the question, you gave me the answer. Thank you. So, what's the question says? I have to read it. In native phase, the proteins separate depending on their size, charge, pi, all of the above. And what have you said? A. It is saying in native page. It is not an SDS page. You should read the questions very clearly and properly, because these are the small questions, easy questions, where mistakes happen when you don't read the question. It is telling native page, you do not have any SDS over there, so you can separate the proteins depending on their size. You can also separate them based on their pi, because the pi plays an important role when there is no charge factor playing a role, and you are not providing SDS or CTAB anything, because of which the proteins would be getting a charge. So you can separate them based on all of these three. So all of the above are correct. So A, B, C all are correct. So it's option D. So 13D is the right option, because it's a native page, not an SDS page. You are not giving any charge to your protein. It is a protein in the native form, the form in which it loves to stay. So it can separate via your size, charge, and it does not even have a uniform charge to mass ratio. So you can separate them based on its PI as well. Okay. Moving to the next question. Number 14 says the pore size of the polyacrylamide gel can be changed by changing the acrylamide concentration, changing the bisacrylamide concentration, changing the ratio of acrylamide and bisacrylamide, all of the above. So how can you change the pore size? No. Even the cross-linking concentration is also important. I was speaking of percentage T and percentage C. So the your cross-linker concentration also plays a role over there, as well as the ratio of the concentrations which you are using. That is also very important. You have to change the acrylamide concentration. It will change the pore size. The bisacrylamide concentration will also hamper your pore size, as well as the ratio what you are using will also. When you see these chemicals coming, if you have seen these Biorad bottles or this SRL company, these acrylamide and the base acrylamide, you will be seeing that there is a ratio given 30 is to 1, 40 is to 1, and these are having different concentrations of your acrylamide, base acrylamide, and also the ratio changing that will also affect it. So again, all of the above statements are correct. So 14D is the right option. Moving to question number 15. In electrophoresis, DNA will migrate towards. This is very easy. DNA is negatively charged and anode is positively charged. Yes, it will migrate towards the anode. These are small things you need to check. Okay. So 15A is right. DNA will migrate towards the anode. To reduce the pore size of agarose gel, one needs to. What do you need to do to increase the pore size of an agarose gel? Increase agarose percentage, decrease agarose percentage, increase salt content, change the pH of the buffer. What would you do? So when does the pore size decrease? So reducing the pore size means having what? How high or how low is the concentration of your agarose? So basically it would depend on your agarose percentage only. So a higher pore size means what? Yes, 16A is right option. Increasing the agarose percentage will actually reduce your pore size of the agarose gel. Okay. To visualize DNA in a gel, a common dye is. What is a common dye to visualize DNA in a gel? Yes, ethidium bromide is the right option because ethidium bromide intercalates within your DNA strands and actually fluoresces under the UV transilluminator. Right. Question number 18. In agarose gel, the migration of DNA depends on. What does the migration? So it is a MSQ question. You have one or more than one option correct. B and C. Net charge, we are speaking of DNA has a uniform charge. So when we are telling of, okay, the migration of DNA net charge, the DNA will migrate towards the negative, towards your positive, that is towards your anode, that is known to us. But the migration of the DNA, of the different fragments of DNA, what will it depend on? Size is correct. Agarose concentration of the gel, it is not correct because that will actually hamper the movement of all the fragments. Okay, not a particular fragment. So if it is not hampering up the two mixtures or the two different fragments, then we cannot call that it is actually involved in the separation process. So the migration of the DNA will depend on its size as well as its shape, because if it is a circular, I haven't spoken about it though, but if it is a circular, say we are talking of a plasmid, if it is a circular plasmid, it moves slow, so the size matters. But if it is a linear plasmid, it moves faster. Even when they are of the same size, if they have different shapes, their movement won't be same. So depending on the size as well as shape, both are actually important for your migration of the DNA. The charge is not important because it has, DNA is negatively charged and move towards the anode, and the agarose concentration will affect all the fragments, not a particular fragment. Now coming to something interesting. Which of the following statements do you think is correct? In SDS page, all the proteins are negatively charged. In native page, proteins can be negatively charged, positively charged, or neutral. In agarose gel, nucleic acids are positively charged. No denaturing agent is added in agarose gel electrophoresis. Which of the following statements is or are correct? Read the question carefully before answering. You would be getting the right answer. So you can expect a few MSQs in your assignments as well as in your exams as well. Okay, I've got an answer, right? A and D. So option A is correct because in the SDS page all the proteins are negatively charged because the SDS imparts uniform negative charge to the proteins. That is known to us. Option D is also true because denaturing agents are not required for your analyzation or analyzing your nucleic acids by running the agarose gel electrophoresis. How about option C and B? So option C says nucleic acids are positively charged. This is false because nucleic acids are negatively charged due to the presence of the phosphate backbone. How about option B? It speaks of the native page. So native page does not contain SDS. So the charge of the protein will be depending on the pH of the protein in relation to the pH of the buffers which we are using. So your proteins can be any charged depending on what pH of the buffer you use and the pH of the proteins as well. So here option A, B, D, three are correct. Because in SDS page you know it is correct, D you know it is correct, but B it is a native page. That is your proteins are in native form. It can have any charge, right? Okay, so A, B, D are correct for question 19. So last question, question 20. Which of the following statements are true? A polyacrylamide gel can be depolymerized by heating. An agarose gel can be depolymerized by heating. In the absence of bisacrylamide, acrylamide forms a linear polymer. It is easier to control pore size in polyacrylamide gels than agarose gels. We have got two interesting questions in the end. That's good. So these are the questions which you can definitely expect. B and C. Okay. So option A is incorrect because in the while speaking of the characteristics of your polyacrylamide gel, I was speaking that they cannot be depolymerized and reused by heating. So B option A is in any ways eliminated. However, your option B is true as the polymerized agarose gel can be reheated to melt it and recast to be reused. So B is right. It can be depolymerized by heating. Option C is also correct because bisacrylamide forms crosslinks. So in the absence of these, acrylamide will only be forming a linear kind of a polymer. That is also true. How about option D? It states it's easier to control pore size in polyacrylamide gels than agarose gels. So, in the polyacrylamide gel electrophoresis, there are two variables with which we can regulate the pore size. One is the ratio of the acrylamide to the base acrylamide as well as the concentration of the acrylamide and the concentration of the base acrylamide. But in the case of agarose gels, only agarose concentration can be adjusted for varying the pore size. So, what do you say? Is it easier to control the pore size in polyacrylamide gels or no? Because in agarose gels, just by only adjusting the agarose concentration, you can actually modulate your pore size. But for polyacrylamide gels, you you need to actually check the ratio of the polyacrylamide gel, your acrylamide to base acrylamide as well as the concentration of your acrylamide and the concentration of your base acrylamide. So it seems to be a bit more complex than your agarose gel. So agarose gel is simpler. So yes, option B and C are correct. Okay, that's all then. I am done with my part. Thank you. If you people do not have questions, then we can end over here. Okay, thank you.